So this is a continuation of Chapter 3 Notes, Part 3. So this is Chapter 3 Notes, Part 3.2, if you will. So I just talked about the neuromuscular junction and the fact that you have the electrical stimulus coming from the nerve cell that needs to get to the muscle cell, and the two of them are not connected because we have that synapse or synaptic cleft in between the two. And so what can help facilitate get that electrical current or action potential from the nerve cell to the muscle cell is the neurotransmitter, which in this case for skeletal muscle cells is acetylcholine. Acetylcholine is going to be released via exocytosis from the axon terminal, and it's going to bond onto ligand-gated sodium channels, which is going to cause sodium to rush into the cell. And that sodium rushing into the cell is going to disrupt that polarized nature of that resting skeletal muscle cell and cause it in that local area, the neuromuscular junction, to become depolarized. Once the threshold potential, it goes from -70 to +30 millivolts, is reached, then voltage-gated sodium channels throughout the sarcolemma and T-tubules are going to open, and we'll talk about that further. So talking about ACH coming down here, binding to these ligand-gated sodium channels, sodium rushes in, we reach the threshold potential, then we have voltage-gated sodium channels that open. And something that you could potentially draw here, let me get a little pen, I'm going to draw it over here. So you have the transverse tubules that are coming in like this from the sarcolemma. And so you've got this essentially electrical wave that's happening, just like we saw up here, that's happening through the sarcolemma. So this electrical wave with these voltage-gated sodium channels comes down the T-tubule. And let's pretend in here, this is a myofibril, okay? And then on top of that myofibril, we have, the Sarcoplasmic Reticulum here. I kind of draw the two ends. The Sarcoplasmic Reticulum, that stores calcium. Well, when this action potential comes in via the transverse tubules, it stimulates voltage-gated calcium channels to open, okay, which cause calcium to be released into the sarcoplasm, which is the cytoplasm of a skeletal muscle cell. And I've mentioned it a couple times, but calcium is essentially the final go signal for the sarcomere to contract. I know that seems like 5.5 years ago, but the whole purpose for this happening is so that the sarcomeres can be stimulated, contract, so that myosin can grab onto actin and cause that sarcomere to contract, which will cause the overall muscle cell to contract. So when a nerve stimulus reaches the sarcolemma, what's going to happen is that ATP is going to fuel the myosin heads to grab onto actin. But myosin can only grab onto actin if calcium is bound to actin. What calcium does is that it actually ties up bodyguards that have been hanging on to actin and allows the ATP binding sites on actin, I'm sorry, not ATP, the myosin binding sites on actin to be free, okay? So the two molecules that are required for the sarcomere to contract are ATP and calcium. And again, that contraction is when the myosin heads reach up, grab onto actin, and pull them towards the end line so that the H zone disappears. ATP is going to bind onto myosin. It's going to give it the energy for those heads to reach up, grab calcium, grab actin and pull them towards the M line. Calcium, all right, is going to bind onto actin, and it's going to, there's two different bodyguards that are essentially hanging on to actin, preventing myosin from binding on it. They're troponin and tropomyosin. And calcium basically comes over and it's like, hey, how's it going, guys? And the troponin and tropomyosin forget to, the binding of it changes its conformation. And now the ATP binding sites become available and myosin can grab onto it. So what the heck did I just say? So again, we've got our, I'm going to switch colors. I'm going to go to a blue. I think this might be easier to see. So remember, a sarcomere is from Z disc to Z disc, and this green here is showing actin, and the red is myosin. And so what happens, all right, I'm going to, so here we have your, let's say this is the T-tubule coming down here, and then we have our grandma's holy sweater, our sarcoplasmic reticulum. Every Z disc has one of these structures associated with it. There's 1, 2, 3 components, which is a triad. So it's 1 T-tubule and 2 what we call terminal cisternae of the sarcoplasmic reticulum. And so the T-tubule brings in that electrical current, which causes voltage-gated calcium channels to open in the sarcoplasmic reticulum. And now calcium, so I'm going to draw Ca, can bind on to actin. So ATP, which is down here, is binding onto myosin and causing these myosin heads to basically stand up and be ready to grab on to actin. The calcium comes over and binds onto those 2 molecules I told you, that troponin and tropomyosin, which are the bodyguards for the myosin binding sites on actin. And the binding of calcium onto actin causes it to change its conformation. And now this head can bind on to actin and pull it. So again, remember, actin is going to get pulled this way. It's going to get pulled this way, and the H zone is going to disappear. So the 2 molecules that are needed, ATP and calcium. So again, calcium comes from the sarcoplasmic reticulum. ATP comes from the mitochondria that are in the cell, and we talked about how there was an abundance of ATP, an abundance of mitochondria. They're going to be producing ATP inside the skeletal muscle cell. There are actually 2 other places that skeletal muscle cells get ATP from, and that's going to be on the next presentation, but your skeletal muscle cells generate and need and use a lot of ATP and oxygen, okay? If there's not enough oxygen, okay, to go through aerobic respiration, then anaerobic respiration is going to happen, and we've talked about this. So with aerobic respiration, we've got oxygen plus glucose will end up making 36 ATP. However, if there's not enough O2, so it's not in big enough supply, glucose is going to generate 2 ATP. So that's quite a big yield difference. And so when we get into anaerobic conditions, we're going to be burning a lot more glucose to make ATP. And a byproduct of anaerobic glycolysis is the buildup of lactic acid, which can cause muscle fatigue and soreness. And this is just showing, so this brown thing here is the mitochondria. And glycolysis is actually the first step of ATP formation that happens out in the cytoplasm, or in this case, we're talking about the sarcoplasm. And it's the first step, and if there's enough oxygen, then the process is going to continue on inside the mitochondria for aerobic respiration. But if it's not, it's just going to keep going through this cycle right here and get that really low yield of ATP and also that buildup of lactic acid. So this picture, so again, we would see the action potential coming down the end of the axon to the axon terminal. This action potential is going to stimulate those voltage-gated calcium channels to open. Calcium is going to rush into the axon terminal. It's going to cause the neurotransmitter to be exocytosed into the synapse. The neurotransmitter acetylcholine is going to move down its concentration gradient and bind onto ligand-gated sodium channels on the sarcolemma, or plasma membrane. Sodium, Na plus, is now going to rush into the skeletal muscle cell, but only here, only in this area, in the neuromuscular junction, because of those ligand-gated channels that were open. And we're going to go from negative 70, and it's only going to happen, so it has to reach this magic number, which we call the threshold potential, which we're saying is 30, positive 30 millivolts. So once this area reaches positive 30 millivolts, then voltage-gated sodium channels are going to open. And that's really an instantaneous thing. And now we have this current that's going to be brought all the way through the sarcolemma and down the transverse tubule. We know that, and this picture is showing it over here. So here we have the transverse tubule bringing the electrical current or action potential down. And it's going to stimulate the sarcoplasmic reticulum, which is right here, to stimulate calcium. Calcium is going to come out, all right, into the sarcoplasm, and it's going to be able to bind onto actin. Now, myosin's had that ATP there. It's been ready. It's been activated, but the bodyguards have been blocking those myosin binding sites on the actin. And so calcium comes over. It binds to the troponin and tropomyosin, changes its conformation. The myosin binding sites on actin are open, and now actin can be pulled towards the M line, and the H zone can disappear. And I told you that these two terminal cisternae, we call them, so there's 1, 2, and then this would be the T-tubule. These three things together, we call them a triad. And every Z-disc has a triad sitting on top of it. And basically, what's going to happen is that the sarcolemma has to return back from, so it was polarized, then it became depolarized. Well, before another action potential can come again and affect this cell, it has to be repolarized. So we're going to go from negative 30 until we reach, positive 30 till we reach positive 45. And then this positive 45 is going to cause voltage-gated potassium channels to open, and potassium is going to rush out of the cell, and potassium ions have a positive charge on it, and that is going to repolarize the cell. So we're going from polarized to depolarized to repolarized. And this is basically talking through those steps. All right, so an action potential being generated is going to be propagated or sent across the sarcolemma down the T-tubules. Is going to cause the sarcoplasmic reticulum to release calcium. Calcium is going to come over and bind onto actin. It's going to expose the myosin binding sites. Myosin has been activated by ATP to come over and grab onto actin and pull it towards the M line so that the H zone can disappear. This is going to happen for a certain cycle of time. Acetylcholine is going to end up being broken down by an enzyme. What's going to happen then is that potassium is going to rush out of the cell. These calcium channels are going to close. Grandma's going to vacuum up all the calcium again, and this process is going to stop, and it's going to basically reset to happen all over again.